Hi all, in this lecture we'll speak about the next type of your staining method, that is your differential staining method. So this differential staining method is used to differentiate microorganisms into two categories based on the staining method they are being responding to. So the two types of your differential staining methods can be your gram staining and your acid-fast staining. So the gram staining is performed widely for differentiating more than all the groups of microorganisms, and the acid-fast staining is specifically used to differentiate microorganisms to check their acid fastness, to differentiate your mycobacterium and non-mycobacterium species as well. Speaking about your gram staining. So this gram staining was discovered by the scientist named Hans Christian Gram. In order to honor him, the name gram staining was given to this technique. So this gram staining method is used to differentiate bacteria into two basic categories, that is your gram positive bacteria and gram negative bacteria, which is solely based on the composition of their cell walls respectively. Speaking about the bacterial cell wall structure, as we have already discussed, we have two different groups of bacteria respectively, your gram positive and your gram negative bacteria. So your gram positive bacteria has a thicker peptidoglycan as compared to your gram negative bacteria. So your gram positive bacteria have 80% of your peptidoglycan layer, but in contrast your gram negative has only 10 to 20% of the peptidoglycan layer. In addition to your peptidoglycan layer, in gram-negative bacteria, we have another membrane outer to your peptidoglycan region called as the outer membrane layer is seen, which is absent in case of your gram-positive bacteria. So this primary differentiation is seen in case of your gram-negative and in your gram-positive bacteria. So this gram-negative bacteria's outer membrane region is made of three different types of components, that is your lipopolysaccharides, lipid A, and your porin membrane, which imparts your pathogenicity to the gram negative bacteria than your gram positive bacteria. So the gram positive bacteria's peptidoglycan region is made up of a specific acid called as your teichoic acid, which gives additional stability, and it replaces your gram negative outer membrane region and imparts your extra stability to your gram positive bacteria. Before moving on into the procedure for your gram staining, we will now discuss the basic principle behind your gram staining procedure. So as we have already discussed, the gram staining helps in differentiating the microorganisms, or specifically the bacteria, into two different groups, that is your gram positive and your gram negative bacteria. So first, the primary stain is taken up by both groups of your microorganisms, both your gram positive and your gram negative, and upon your alcohol treatment, due to the higher amount of your lipid layer present in case of your gram negative bacteria, these lipid layers gets hydrolyzed or washed away by your alcohol, and so the primary stain gets washed off in case of your gram negative. But upon your alcohol treatment, there is less amount of your lipid content being present in case of your gram positive bacteria, and so the primary stain is retained till the end of your process and your gram positive appears purple in color. But in case of your gram negative, as it is colorless upon your alcohol treatment, we now add your counter stain or your secondary stain, which are absorbed by your gram negative bacteria. As a result, your gram negative appears pink in color, and so this staining technique is called as a differential staining technique, as these two groups of microorganisms are differentiated based on the color or the dye they observe. As a result, your gram positive bacteria appears purple in color and your gram negative bacteria appears pink in color. Now we'll move into the procedure. First, make a thin smear of your culture on your slide. Heat fix it with the help of your Bunsen burner. Now stain the smear with your primary stain, that is your crystal violet, for 1 minute. Next, here we now have the formation, or we now have the absorption of your primary stain by the microorganism. Next, wash your slide with the running tap water. After washing, add your grams iodine as a mordant or your fixative, which helps in fixing your primary stain to the cells for 1 minute. Next, rinse it with water. Now add your decolorizer, that is your alcohol, for 15 to 20 seconds. Now stain it with saffronin, which is your secondary stain or your counter stain. Wash it, blot dry or air dry it, and then observe under your microscope with oil. So these are some of the images that are observed under your microscope upon your gram staining. So the first image shows your gram negative bacilli, the second image shows your gram positive bacilli, and the third image at the bottom shows your gram positive bacteria, specifically your cocci under clusters, that is your staphylococcus. Next type of differential staining technique we remove is your acid fast staining. So it was first identified and discovered by Ziehl-Neelsen's, and so this type of staining technique is also called as your Ziehl-Neelsen staining. So this differential staining method is used to differentiate bacteria into two categories, that is your acid fast and non-acid fast bacteria. So the acid fast bacteria are, they resist your decolorization mechanism upon your acid or alcohol treatment due to the presence of a specific layer called as your mycolic acid layer in their cell wall. So this mycolic acid layer is a specialized layer which helps in preventing or resisting the microorganisms for your acid alcohol treatment, and it is difficult for the microorganisms to be eradicated due to the presence of mycolic acid layer, as the antibiotics do not penetrate inside your cell membrane of the microorganism. It imparts your additional stability and strength against your particular antibiotics, and so this group of microorganisms, that is your acid fast microorganisms, are life threatening. This mycolic acid layer is responsible for the slower growth of your microorganism, as the nutrient uptake is difficult from the environment due to the presence of this mycolic acid layer, and so the generation time of these microorganisms are longer. Approximately 14 to 15 hours is required for the division of one cell to be into two. So the example for your acid fast bacteria is your Mycobacterium species, that is your Mycobacterium tuberculosis, Mycobacterium bovis, etc. And your non-acid fast bacteria's example is your Streptococcus pneumoniae and your Bacillus subtilis. We'll move into the procedure for your acid fast staining. So first, smear the culture on the slide. So it should be an even thin smear which helps for the even distribution of the microorganism on your slide. Next, application of your carbol as your primary staining for 2 minutes. Now apply heat for 5 minutes, which helps in the easy penetration of your primary stain into your cells due to the presence of this mycolic acid layer. The application of your heat for a longer period helps in breaking down the mycolic acid layer, and so the easy penetration of your primary stain takes place and the cells get stained. Now decolorize the smear using 20% H2SO4, that is your sulfuric acid, or 3% of your hydrochloric acid, or 95% of your ethanol for 2 to 3 minutes. If the bacteria is non-acid fast, they decolorize and they appear colorless. If it is an acid fast bacteria, this decolorizing treatment is resisted, and so the primary stain is retained by the acid fast bacteria. Next, stain it with your counter stain or your secondary stain. It could be your methylene blue. Next, wash the slide, blot or air dry it, and observe under your microscope. So the primary stain is carbol and the secondary stain is methylene blue. Other than your methylene blue, there are many other secondary stains or your counter stains can be used. That is your picric acids can be used and malachite green can be used. So your non-acid fast bacteria appears blue in color if methylene blue is used. It could be green if malachite green is used, and it can also be yellow in color when your picric acid is used as your as your secondary stain. As a result, the image shows positive for your mycobacterium's presence, as the carbol color is observed by your acid fast bacteria and appears bright red in color against your blue color background.